everyone. We all had been discussing till now about the enzymes which were required for the process of replication. Now, let's look up to the entire process of replication or the steps of replication. The very first initiating step is initiation. In this, the replication fork formation takes place. Replication fork is an opening which is formed at the origin of replication. Origin of replication is a site over where the process of replication will actually occur. Now, not only that, this origin of replication is the site where several enzymes go and bind up. These all enzymes which have went there and have binded, they include the helicase, which helps in unwinding of the DNA strands, the RNA primer, which is synthesized by primase enzyme, and it is 10 to 12 nucleotide long and helps in initiating the process of replication. Then, after the DNA polymerase starts the entire process of replication on both the leading as well as on the lagging strand. Now, the leading strand is the strand on which the direction for the DNA synthesis is 5' to 3' and it is continuous. So, with the help of the DNA polymerase, at first this 1 5' to 3 in 5' to 3' direction, one entire nucleotide strand is formed. Whereas in case of lagging strand, small chunks of RNA primers are added and then after the DNA is formed. Again, small chunk of RNA primer will be added and as a result DNA will be formed. And in this way, these RNA primers they need to be removed off once the DNA process is completed or DNA replication process is completed. So the removal of this DNA, these primers will create gaps in the DNA structure which is formed, and these gaps need to be joined with the help of DNA ligase. So this is how the initiation occurs. Basically, the most important part about DNA first step, DNA replication's first step is recognition of ORI, which is origin of replication, addition of helicase, and these all other enzymes like topoisomerase, SSB, and all, in order to make the process much more convenient for the replication to occur. Now, as we proceed, we look into this diagram. You all can see that in this diagram there is presence of uh, open DNA structure, and not only that, you can see some of the other structures which are mentioned. In the center, there is presence of a origin. Every time the DNA synthesis which is taking place, it is taking place in 5' to 3' direction. This one is 5' and this is 3'. So this is a continuous strand because you can see there is no gap in this strand which is formed. And this strand which we are talking about is the leading strand. Similarly, if this is the site of origin, then this 5' to 3' direction is also the replica of continuous DNA replication. And not only that, it also helps in forming the new strand continuously with the help of DNA polymerase. Once these leading strands are formed, during the same time, the lagging strands are also getting formed. If you see the structure of lagging strand, all the red color portions are the lagging strand DNA nucleotides which are formed, whereas the green ones are RNA primers which need to be removed once the entire process is completed. But why are the DNA molecule or why the DNA molecule is not getting synthesized continuously, but there is gap in between them, or every time there is addition of little amount of RNA primer in order to initiate the process? It is so because as soon as the helix opens, at the base there is addition of small amount of RNA primer, and as because the direction will always be from 5' to 3', so the DNA primer will be formed from 5' to 3', and just as soon as it reaches the 3' and it will stop. And this is how the entire process continues. And once the DNA is discontinuously formed, we'll And if we look up to the entire structure, it looks as if there are small portions of DNA formed with the help of RNA. And there should be removal of all the RNA subunits. Once the RNA subunits are removed, then again DNA polymerase runs over it in order to form the DNA nucleotides. So, initially, the very first step is for this entire process of DNA replication. First thing which we require is the finding out, or we need to figure out that which portion is known as the origin of replication. Second important thing is once the ORI is found out, we need the information about activation of deoxyribonucleic acids, because these are the substances which will be providing us energy in order to perform the hydrogen bonds or in order to form the dihydrogen bonds. Most of the basically what happens during this process is DNP, all the monophosphates like deoxyribonucleic deoxyguanosine monophosphate, it gets converted into deoxyguanosine triphosphate. And in the similar way, adenosine monophosphate, cytosine monophosphate will also get converted into triphosphate. When and this conversion takes place with the help of phosphorylase. And with the help of phosphorylase, where this conversion is taking place, one thing is to be kept in mind that triphosphate, the function is every time the phosphate molecules will be released, they will be releasing enormous amount of energy. With the release of enormous amount of energy, we will have the production of, or this conversion basically occurs in presence of phosphorylase enzyme, that is to be kept in mind, addition of phosphate in that way. Okay, next is about the opening of DNA helix. Once, how does this opening occurs, or how is it initiated? With the help of these few enzymes functions you are already aware of. So let's move on. If you see the structure, you can see at first the helicase will come and bind and will unwind the double helical structure of DNA. Then after topoisomerase will come and get attached to the double helical structure of DNA and reduce the tension. SSB helps in topoisomerase. One more important thing is it cuts and twists the DNA strand and reseals it, because when we think about the DNA double helical structure, there are two kinds of turn. One is a circular turn, and the other another one is uh, just twisting amongst both the strand. So twisting can be removed, but the circular turn needs to be stabilized only after cutting and twisting and resealing it. So that is what we do. After that, the SSB protein comes and binds in between all both the strands so that they do not single strand stabilizing enzyme. After binding of these enzymes, the both the strands they lose the capability of overlapping with each other, and because of that DNA polymerase can act over the RNA primer and synthesize the DNA structure. This is about the initiation of the DNA replication. What happens in the elongation process? Continuously new strands are getting formed in the leading as well as on the lagging strand. Leading strand, the formation of DNA nucleotides is continuous. So you can see that in this structure, a continuous loop is formed, or continuous strand is formed in 5 prime to 3 prime direction. Whereas in the other one, you can see these few gaps formed. Other gaps are definitely there, but they all are resealed with the help of ligase and DNA polymerase. So once these strands are formed, then after the gaps will be there, and these all gaps are filled, known as the Okazaki fragments. Okazaki fragments are as such areas which help in the uh, or they are named as such because of the presence of this continuous small chunks of DNA formation. and that's why they are a little weird in, they look little different than the normal DNA structure. So this is important. Next is the same diagram in a little more elaborated way. Every time when the small nucleotides, or whenever the nucleotides are getting attached, few things are to be kept in mind. First is that the RNA and DNA bonding takes place at a very less time. That's why we use the RNA primer. And when the new nucleotides come, they have two sites for attachment. One is the DNA and another one is the RNA primer. So the RNA primer helps in holding the DNA at a very fast speed. So with the help of this, the entire process takes place in a little rapid way. If you see this structure, you can see the single stranded binding protein over here. Helicase is binded at the parental strand. Primase, as soon as the primase is bound, you can see the red color, or the red one, small chunk of 10 to 12 nucleotide long structure, and that is formed with the help of primase. This red portions which are formed, they all will be removed once the entire DNA strand is formed. And this is what happens throughout the process. DNA polymerase 1 is helping in removal of the primer by, and DNA polymerase 1, the function is this only, in order to remove the RNA primer and form the, and with the help of DNA polymerase form the normal nucleotide sequence, because already half of the sequence is formed. So these all things are to be kept in mind while thinking about the process of DNA replication. Synthesis of leading and lagging strands, it is discontinuous. The lagging strand is formed in a discontinuous way and it takes more time as compared to the leading strand. Once the DNA structure is formed, then the important thing comes, that is proofreading. So the entire, over here you can see in the proofreading occurs continuously when this entire process has been taking place, or DNA replication is already occurring in the lagging strand. At that time what happens is the proofreading portions, or just the DNA polymerase runs over the entire uh, strand of DNA which is synthesized, and tries to figure out that where there is a wrong nucleotide placed. And once it is found, then it is removed, and again DNA polymerase runs over it in order to form the correct nucleotide, or attach it to the correct nucleotide. This is known as proofreading. Proofreading is identification of the wrong nucleotide, DNA nucleotide, which is placed, and repair is removal of that with the help of nuclease activity and addition of the correct nucleotide at the same place. So this is about the repair, and the epsilon unit of DNA polymerase helps in this process of repair. Last is the termination step, over where DNA polymerase halts. Where does it stop the process of DNA replication? It halts when it reaches a section of DNA template where that has already been replicated. So, however, the DNA polymerase cannot catalyze the formation of phosphodiester bond between the two segments of DNA strands, and it drops off. So, as soon as it will reach a sequence where complete synthesis has already occurred, at that time the DNA polymerase which is attached, it will fall off, and that is what happens. As soon as it falls off, the sugar phosphate bonds is already full replicated and it has formed nicks, and now the RNA primers which are there, they all need to be removed and replaced with the DNA subunits, and the sugar phosphate backbone is to be connected with it. So this is how the DNA replication comes to an end, and we all, so I hope it is like little clear to everyone. Here, one thing is to be kept in mind, that magnesium ions are very important for the entire process of DNA replication. They act in initiating the entire process in a rapid way. Okay, thank you very much.